Begin by acknowledging the traditional owners of the land on which we meet today, and I would also like to pay my respects to elders past, present, and emerging. Good afternoon, everyone, and welcome to the April FightMND Scientific Seminar. My name is Dr. Sarah Bennett, and I'm the Senior Research Coordinator at FightMND. We hold these seminars monthly on the last Tuesday of the month, and last month in March, we heard from Professor Michael Dobby, who provided us with an excellent overview of the NCRIS network and specifically the capabilities of Phenomics Australia in supporting M&D research. Associate Professor Trajano also outlined a vision for a national stem cell resource leveraging the capabilities of Phenomics Australia facilities. If you're interested in this presentation and weren't able to join us live, a recording is now available on our website for you to catch up at a convenient time. So following the talk today, we are leaving plenty of time for discussion and questions. So if you would like to ask anything, please use the reaction button to raise your hand, and one of our team will enable your microphone and camera and let you know to ask your question. Alternatively, you can also type your question into the chat, where our team will be monitoring and read out questions on your behalf. Please note that the chat function is only enabled if you're using the app-based version of Teams, and unfortunately doesn't work in the web-based version. So please ensure you do introduce yourself and where you're from before asking your question so we can all get to know each other. So now to our presentation for today. Gabriel Trajano is an Associate Professor in Exercise Science and Neuroscience at the Queensland University of Technology in Brisbane. His research group investigates motor unit physiology, focusing on various aspects of how motor commands are carried by neurons, how this degenerates with age and disease, and how exercise may counteract these changes. Associate Professor Trajano has also been developing non-invasive biomarkers of MND through the application of high-density surface EMG decomposition, a process which sounds complicated to me right now, but I'm sure he will explain it to us in the talk today. Thank you so much, Gabriel, for taking the time to present to the MND research community today, and over to you.
Thank you very much, Sarah. Good morning to everyone. So thanks for inviting me to be here today. I assume you all can see my slides now. So if you can't, just let me know. Yes, good.
Yeah, you're good.
So today I'm going to talk a little bit about high-density EMG and how I think that can be used as a window into motor neuron physiology in motor neuron disease. So first, I will talk a little bit about what is high-density EMG. Then second, I'll briefly touch into motor neuron physiology and PICs, which stands for persistent meter currents, which is one of the main things we try to study. Then I will present some of the results from our FightMND funded project. And then I'll talk a little bit about future directions, where I think things should go and what are ideas for future things. And maybe that could help people to kind of have some ideas and bring some discussions where we can collaborate with others in the future. So the first question is really like, what is high-density surface EMG? So it is a non-invasive technique. It is painless. It has really good clinical applications. So we use surface grids of electrodes that could be 64 or 128 channel grid of electrodes. So actually these now people can use up to 256 electrodes in a grid. It has great high spatial resolution, so it's able to capture the action potential propagation across the skin. With that type of information and signal, we can then use an algorithm that does something we call blind source separation. So it's an advanced algorithm that can then go and decode the raw signals that are the EMG into individual spike trains of motor units, as you can see here in figure 3. So that will yield a large number of motor units. And then you can look at those spike trains of these motor units. So this process altogether, you can see in figure 1 what an electrode grid looks like that plays over the tibialis anterior muscle, which is the typically one we used. We then get that signals, the signal sources from 64 or more channels together, put into a computer, use an algorithm. That algorithm can then, separate those signals and get us spike trains of this activity of single motor units. So in this example here, we can see the activity of up to 16 more neurons or more units firing at the same time as someone completes a task where it tries to increase force and then decrease force. You can see the motor units being recruited and de-recruited. So each different color is a different motor unit. So the very important thing in the novelty is that because you use a high density surface grid of electrode, then we can get the typical signal of a EMG, which is this very convoluted, like signal that doesn't really tell us a lot. And then the blind source separation algorithm can unmix that signal into the spike trains of single motor units, right? So now instead of having this global measure of electroactivity of a muscle, we can specifically go and look into the activity of each individual motor unit, which give us a lot more information. So each one of those bars here, it would represent the firing event of a single motor unit. So each color would be one motor unit firing. So the algorithm does that automatically, which really changed the landscape on how we can research and understand motor neuron physiology. Because in the past, we used to use intramuscular EMG, which is invasive, which is a needle or firewire that goes inside of a muscle, and that would give you recordings of a single motor unit. It's very selective, but it only gives you very few motor units. Now we can use this dense grid of electrodes, which the composition is automatic, it's non-invasive, and then can give us what we call a population perspective of motor units. So it shifts us from solos to symphonies. We would before only have the information of one motor unit. Now we can see how they all work together, how they all talk to each other, how they all behave in the same way or not. So it gives us a much better perspective in trying to understand the central nervous system. So just trying to put everything together. So when we think about EMG, we have different modalities. So there we have invasive or needle EMG. That could also be fine wire EMG. It's very selective and gives just a very few number of motor units for us to study. The normal surface EMG, it's non-invasive. It's just a bipolar couple of electrodes. It really just gives us the muscle activation kind of estimate of a muscle. We can't track motor units from that. And that's probably what a lot of people have seen in the past too. With now these advance in technology for EMG signal acquisition, we can use high density EMG that's also non-invasive. We just stick to the surface of a muscle. There are this dense grid of electrodes, has great spatial resolution, and we get better with more electrodes, and then can yield a very high number, up to dozen of motor units. So I think we have cases we have been able to record more than 40 motor units at the same time. Great, but then the next step, now we know that we can use high density EMG to record motor units. We need to maybe go a little bit and understand what a motor unit is. So the motor unit really is the final common pathway of your central nervous system when it tries to control movement. So it is one motor neuron and all the muscle fibers that innervates it, right? It integrates all the sources of synaptic inputs. So your brain sends your motor neurons, for your spinal cord sends your motor neurons with all the information that comes from the central nervous system, gets integrated at the motor neuron level, and that gets transmitted to the muscle fibers, and that translates into mechanical force. So in the end of the day, when you need to perform, muscle contractions or movement or activities of daily living, that's really the final product of your brain activating those muscle fibers. Very important, when we try to create muscle contractions or movement, we have a very specific way in how we control these motor units. Like there are two strategies that happen in parallel. We can recruit more motor units. So with more motor units, we can produce more force. And we follow what we call Hahnemann size principle, that we recruit first the smaller units, and as the force increases, or we need to produce more force, we start to recruit larger motor units. So these are lower threshold units are smaller, and the higher threshold units are larger, faster, and stronger. The other way we can do it is also by increasing the discharge rate of these motor units. So after we recruit these units, we can ask them, or your brain or your central nervous system can drive these units faster. And if they fire faster, they're able to produce more force, right? Those two things happen in parallel, right? So we increase force by doing the two things at the same time. More specifically, at the very end of the spectrum to the maximum level of force, we rely more on rate coding, on the discharge rates of the units to produce force. And that's very important because we need to understand and acknowledge that everything we do in terms of movement, it's relative to our capacity of create or generate muscle force, right? So if you have a reduced capacity to create or generate muscle force, then our ability to do everything else might also be reduced. So what's special about a motor unit? I think there are a lot of things that are special about motor unit, but one thing that I think it's really important is there is this one-to-one relationship between a motor unit and a motor neuron. So if you record the action potentials of motor units, those are exactly what's happening with the motor neuron that innervate that unit, right? So because the transmission across the neuromuscular junction, it's very safe. The muscle fibers, they amplify the very tiny signal that comes from your motor neuron. So you can read that action potential. And that's really a one-to-one relationship between muscle action potential and motor neuron action potential. So that really opens a direct window into our central nervous system. So these motor neurons are really the unique cell in our central nervous system that can be easily read from the surface, right? From in day-to-day kind of techniques that can be used. So they have a very big clinical value because now we can read how the motor system or the central nervous system is controlling this unit, right? And give us the blueprint of the central command of what your brain, of what type of information your brain, your spinal cord, everything else in the system is sending to these neurons, these motor neurons. They are the final pathway of creating movement or the signals. So we can also, and another important thing is that we can characterize these motor unit populations. So remember, I was telling you in the past, you could only get few of them, but now because we have populations of motor units, have a very large number of them, then we can also characterize them based on their recruitment type. So we have these low threshold units. There are, typically slow twitch, fatigue resistance. They are recruited very early on during low force activities and everyday tasks. Typically, in motor neuron disease, they act more as a compensatory survival. So they go and reinnervate the orphan muscle fibers, while it is evidence then tell us there's higher threshold units. They are fast twitch and highly fatigable. They also require, a high level of activation of your brain, sending like a very large effort to be recruited. Those are the last ones to be recruited. They are only recruited when we try to execute very strong tasks that require high levels of force, or if you really try to have a movement that's supposed to be fast. These are the ones that are supposed to be more vulnerable in MND. Like those are the ones that tends to be degenerate first, possibly because they reduce oxidative capacity. So they seem to degenerate first, and then they get reinnervated by low threshold units, and then they start becoming more like low threshold units rather than high threshold units. So differentiating them can be very helpful in understanding what's happening, because different units, different type of motor units might be going through different types of changes or adaptations through the disease. So you might lose a lot of information if you then put them all into the same bag. So with high density MG and with our recordings, we can differentiate them at some level based on their recruitment thresholds. So this, again, is the same example I showed you before. That's a control participant, like a 60-year-old male. And again, we asked the person to do this task, that we asked them to increase their force up to kind of 40% of their maximum capacity and then decrease their force. As we can see, as force goes up, the more and more units start to being recruited. So these units here in red at the very early part, they are the first ones to be recruited. And later on, you see all these other units being recruited. And at the very end, when force get at higher level, you see all these blue units being recruited, right? So the red units, the reddish colors, are lower threshold. They're being recruited first. And the blue ones are higher threshold units, they're being recruited less, right? You can clearly see how they have a very clear in terms of different recruitment threshold. And as force goes down, they start to being de-recruited too, which is also not as clear as the recruitment is. On your right, you can see an example of a recording from a person with MND. So as you can see, we can get a lot less motor units from that recording. You can see in this example that these low threshold red units are being somewhat preserved, still fire continuously without interruption, while these higher threshold units, they seem to be intermittent failing to fire. So they can't fire as sustainably as the low threshold units do. So they seem to be affected by degeneration. So I like this figure because I think it's a good example of how sometimes you might, if you're just looking at the low threshold units, you might think that there is no problem, but the problem might be specific to a specific population of motor units. Good. So before we move on to talk a little bit more about the specific mechanisms we are investigating, I'd like to take a step back and talk a little bit about how people historically and traditionally thought that motor neurons work, and what's the more contemporary view of how we understand they work. So in the past, people in a lot of the books would not really differentiate this. In the past, people always have this view that then your brain wants to initiate movement and sends a signal that goes all the way, we call it an input, goes all the way to your spinal cord, then it synapses with a motor neuron. Then what gets out is an output that is very similar to the input they got in, right? So the traditional view of what a motor neuron is that motor neurons have this linear input-output relationship. They work as just passive cables. They just relay information from your brain to the periphery. So they have this linear input-output relationship. But since the 80s, when people start to change the experimental models they use on animals, and I can talk more about that later if people need me to, we start to understand that this is really not how it works. So in reality, our brain sends some input to our motor neurons, and I'll send here the same similar input that goes to the motor neuron. The output is not linear. The output can be amplified at a motor neuron level. So the motor neuron produces an output that is larger than the input received. So the motor neurons really act as an amplifier, right? And the amplifiers that we have in our motor neurons, we have other things that would also help as is amplification source, but then one of the main ones is these PICs. So the PICs are our motor neurons amplifiers. So they are short, the abbreviation we use for persistent inward currents. So they are inward or depolarizing currents. They are largely on the dendritic body of the motor neuron, and there are sodium and calcium channels that remain open for a bit longer. So they can be facilitated by monoamines, which are serotonin and noradrenaline. That means in the presence or at higher levels of these monoamines, we would have greater facilitation, right? And these days now we think that this is almost a parallel system that works together with our motor neurons to change the gain of our motor neurons. So more serotonin and more noradrenaline can be released in tasks that require motor neurons to operate at higher levels, and maybe less of it will be released when in tasks that we require the motor neurons to operate at lower kind of intensity levels. So these PICs, they change this input-output relationship so they can increase the gain. And as I just mentioned, we think this gain can be variable. It can be controlled for situations that we want more gain or situations that we want less gain. we can amplify the synaptic input. So the PICs can amplify it up by 5 times. So it can increase the discharge rates of motor neurons up to 5 times. So some computer simulations have suggested that we could only produce, if we didn't have these PICs working and amplifying synaptic input, we could really only produce 40% of your maximal force capacity. So we would be a lot more weaker than we are. So a lot of the tasks that are easy for us to do, it wouldn't be doable if we didn't have these PICs amplifying synaptic input that goes into the motor neurons. So again, this is another diagram just trying to reflect the same information. So in situations that you have the same excitatory input in both conditions, in a situation that has high PIC amplitude, we're going to have a much larger motor output from the motor neurons that would then result in much higher motor unit discharge rates. In situations that have a low PIC amplitude, then a lower motor neuron output and lower discharge rate. So these would be helpful for situations we want high levels of force. These would be helpful for situations we want, precision tasks, for example. So in humans, we try to indirect estimate the contribution of these PICs to the motor neuron firing profiles by looking and try to examining the firing profiles of motor units. So we use these, what we call the paired motor unit technique, and we calculate the delta F. So the delta F is pretty much you pair motor units. We have a low threshold unit that uses that is used to kind of estimate or report the amount of synaptic drive that goes to the muscle. And then we measure the hysteresis into the discharge rates of recruitment and de-recruitment of a test unit. And the difference in that, like a lower de-recruitment comparing to a higher recruitment frequency, is what we call the delta F, right? So it gives us the hysteresis in this recruitment, de-recruitment profile. So what that really means, so a higher delta F, a higher hysteresis, a higher difference here, it tells us there is a greater PIC contribution to this hysteresis. That means you had a more gain in the system. So the amount of synaptic input required to keep the motor neuron firing is lower than the required to start. So it's a non-invasive estimate of this persistent mean recurrence, or at least the contribution to it to self-sustained motor neuron firing. So we've done a lot of work in investigating these. We have, have done many studies in many different populations and in many different conditions. As a summary, I think the important things, we know that these delta Fs decline as we get older. So the PICs will technically decrease as we get older. We also find out that we can use resistance training. So you ask people to lift weights. So we were able to kind of increase these PICs or this delta F in older people. So we saw that was reduced in older people, but we can kind of revert that by asking people to lifting weights. Also, that change or that increase in the delta F was significantly related to the amount of force that people could now produce after training and how much better they got into functional tasks, like, raising from a chair and walking, that was related to that. So which give us some indication that this delta F might also help us to explain the people's ability to perform functional tasks. We also found that these PICs got reduced after you stretch a muscle. So if you stretch a muscle, this tends to go down, which then decreases motor neuron excitability, which might help to explain, why some people think that things like stretching might help to control spasticity, because spasticity is just a motor neuron response and being overactive. So that's why it's possible that stretching might have some beneficial control over spastic muscles. And we also found that these delta F scales with force demand. So in tasks that require higher levels of forces, we see a larger delta F. So these could be contributing for someone's ability to be able to produce a higher motor output. So in conclusion, there is good evidence that this delta F kind of is dynamic. It can change based on different tasks and could be used as a trackable physiological marker. function. But probably the questions you're thinking now is like, are these PICs involved in ALS? So the answer is yeah, absolutely. In animal studies, we know since the early 2000s that people have found the PIC amplitude to increase in presymptomatic SOD1 mice model, but also some animal evidence suggests that at later stages of the disease, then when it becomes symptomatic, then this PIC amplitude tends to reduce, right? So the animal evidence suggests that you might go a period, the initial period of hyperexcitability, and that goes down and decreases those PICs as the disease progresses. So in humans, we published a preliminary study a few years ago where we tried to look at the delta Fs, and we have people coming for 2 visits at the Royal Brisbane Women's Hospital. What we tried to do, we tried to then leverage on the fact that some patients would have muscles that had no signs of clinical weakness, and the same person would also have a muscle that have a strong sign of clinical weakness. So we try to split them into, if have an MRC scale of 5, you say they have no clinical weakness, and they go less than 5, we have some sign of clinical weakness. So what we found is that these delta Fs, which are estimates of PICs, they decrease in muscles that have signs of weakness, and they have increased with muscles that have no signs of clinical weakness yet. Which goes with the theory that, we might see a period of hyperexcitability before the disease progresses further and then goes down. Just an important piece is that this increase we found in this study was really, really small, okay, and was the initial finding. So that makes us think that, tracking changes in delta Fs in people with ALS has the potential to be a relevant clinical biomarker of disease progression. We then, a few years ago, we get funded by FightMND to do this project and look at the delta F as a biomarker of disease progression. So you want to look at the longitudinal changes and try to find out how motor neuron excitability changes over a 1-year period and use high-density MG to track motor units. We also try to look if the measures we are performing, if they would relate to the standard clinical tests, the ALS, FRS scale, the MRC scales. And another aim of that project was to try to separate different types of motor neurons. So you can look at the lower threshold and higher threshold motor neurons and see if they had different patterns of disease progression. Our hypothesis really here was that the high threshold motor neurons would be the ones that would be the most affected. So we had a 12-month kind of longitudinal cohort. Our goal was to try to recruit people at repeated visits over a year period. As typically happens with these types of studies, we have more participants at the beginning and less participants at the end. It doesn't mean only 5 completed the 1-year trial. It means 5 participants were the ones that have 5 visits over a period of 12 months. We might have some participants that might have completed the 1 year with 3 visits, for example. So we have different number of participants that were tested at different time points. So putting all together, we have to kind of work with a statistical model that take that into consideration more of the time rather than the number of visits itself. So these represents the largest longitudinal data set that has used high density EMG in trying to track progression of the disease that I'm aware of. I don't know of any study as large as this. So what we found that, the delta F progressively decline. So there was a steady decrease of around 0.025 hertz per month, which is average to equal 0.3 hertz per year, reflecting our ongoing degeneration of the motor neurons over that period of 1 year. You can see in this kind of red color, what there are the muscles. There are the weak or the MRC less or equal 4, and you have in blue the strong muscles, so muscles that are deemed to be clinically not weak yet. there was no really big difference between how they progress. Both of them seems to go down a long time. The changes in delta F did not correlate with changes in the total ALS FRS score, so there was no correlation between those two tests. So in this graph on your right, you can see these two lines kind of together, so showing a progression because they are not different. When we try to look to the specific results on different types of motor units, so you divide them into three tertiles, so low, mid, and high threshold motor units. What I saw is that the group, and that was very surprising, the group that was contributing the most for the decline was this low threshold motor units. So we can see in this, pink, red color that goes down more than the blue and green color. Okay, so the T1 is the low threshold motor units, which seems to want to be driving the most these reductions. So they showed a decline of around 0.66 hertz per year. So it's also possible that this is the type of motor neurons or motor units that are the most common in our sample. So they must be driving more our results. So in summary, the results we have from the delta F, it shows that there was a progressive decline of these values regardless of clinical measures and regardless if muscles were deemed to be clinically strong or weak. These reductions in motor neuron excitability, we think they kind of mimic or they just follow motor neuron degeneration, so they are closely related to the motor neuron degeneration process. This greater reduction in lower threshold motor units, which was surprising. We were expecting to see a greater decline in high threshold motor units. It could tell us that these low twitch and fatigue resistance motor units were the ones most affected, which I'm not sure if that's the case. I think probably what we're seeing here is motor unit remodeling and the recruitment compression. So these high threshold motor units that used to exist, they don't exist anymore. And once they die, they get reinnervated by low threshold motor units. So the whole recruitment process or range gets compressed to a much narrower range. So now we might see a more biased recruitment towards lower threshold units. And therefore, what we're seeing now at the end of the 1 year period or the second half of it, it's more the remodeling of those higher threshold units that die and get reinnervated by lower threshold ones. And now we measure them and identify them as lower threshold ones now. And that's because we're not able to track these motor units longitudinally. As you understand, the disease changed motor units and it's changed the muscle fibers too. So the shapes of these action potentials will change, which will make it, basically impossible to track motor units longitudinally over a period of a year. So because we can't do that, we can't be sure that we're following the same motor units across the whole year period. So I talked about the delta F results first. So now I'll go into some, I'm calling secondary results. They're not really secondary. I think they're really important results too. So with this test, with the delta F test, before we actually do the delta F test, we had to ask people or the participants to do maximal contraction. So we would know their 100% of EMG, and we therefore could calculate the 40%. So we can use in the delta F test. During that contraction, we were able to record the EMG and then decompose the motor unit activity. And we could then look at, during maximal voluntary contractions, what was happening with their motor units, what happened with the motor neurons, how were they firing? So here you can see an example of a participant being tested at baseline. So first visit, each color represents a single motor unit, and the lines is the discharge profile of them. So the firing rate goes up and goes down at the end, right? So the circle here is the point where you can see the highest discharge rate for each one of those motor units. That's from a single person. The line really reflects the average of the firing of all those units together. So you can see at baseline, you can see a lot of different motor units. They fire kind of regularly at an average discharge rate of 30 hertz. 5 months later, we still can see a large amount of motor units there. The average now for those units is a bit lower. It's below 30 now. And you can see 12 months later, we can identify a much lower number of motor units. So possibly the disease has progressed. A lot of the motor units are gone now. And the average discharge rates of these units and even the peak discharge rates now, like you can see, the highest firing off a discharge rate is less than 30, which is less than the average for all the discharge rates in the baseline. So it gave us a really good picture of the disease progression in a task that they have to perform close to their maximum. So you put all the participants together, what we found is that these maximal motor unit firing rates, they decline significantly over time at a rate of around 0.32 hertz per month. These were also well related to the clinical measures. So they decline as people. So here we look at progression a long time, right? So if we assume that people progress a long time, we could see reductions, but we could also look at progression as they progress through having a lower MRC score. That could be used as a measure of disease progression. And as they progress from a 5 to a 4 to a 3 to a 2 to a 1, we could also see a decrease in these maximum firing rates for each one of them. So these are the same people, and then I try to track them and look at the values as they change their score. So for each 1 point reduction in the MRC scale, we have a 2.4 hertz reduction in the maximum discharge rates capacity. And we also saw a 0.54 hertz reduction for each 1 point reduction of the ALS FRS scale. So these maximum motor units firing rates, they do decline as ALS disease progress. That might be reflecting the surviving motor units that they undergo progressive pathological changes as the motor neuron degenerate, which could be indicating the collateral reinnervation by low threshold motor units. So if the lower threshold motor units are reinnervating the orphans' high threshold motor unit fibers, then they do have a slower motor unit discharge rate profile, and therefore they might be dragging the whole discharge profile down. It could also reflect a reduction in your upper motor neuron to drive the lower motor neuron. So it could be the degeneration of the upper motor neuron part that now cannot drive the lower motor neuron, the spider motor neuron, at the same way they could before. But even if it could, like let's say if the upper motor neuron was exactly the same, it could also reflect that the lower motor neuron, the spinal motor neuron, is now less responsive to the same amount of synaptic input, which would then result in lower discharge rates. So again, with the secondary findings, this seems to be a quite promising tool that could be used as a biomarker of disease progression. We thought that seems to be very promising. It was just preliminary data, and we still have a lot of work to do to improve this as a measurement. So I'm getting close to the end now. So what are the next steps? So what we think we need to go next? So I think one of the things we need to try to do, and we would like to do next, is to improve these measurements. So all those things were we started that 3-4 years ago. So technology has improved, the way we can measure these things has improved. Now we can add force measurements together to our measurements. The technology on signal acquisition, high density EMG has improved, and it can record more motor units. So a lot of the variability we saw in those studies we've done, and we started a while ago, I think it can be reduced now with better methods. So we could be able to improve them even further. So it is possible that we can improve both delta F and maximal discharge rate measurements. We have received a grant from MNDRA to look at intramuscular coherence. So in that one, we're also going to use high density EMG to look how groups of motor neurons fire together. We're going to try to understand how as a population they behave you can use some sort of a mathematical processes to understand the different sources or input sources and how they drive these motor units. And that will tell us a lot of information about cortical spinal abnormalities. And it might be another biomarker that we can work and develop from that. We are hoping to also use this large population of our neurons to investigate interneural networks. So we can use a lot of the paradigms that people have used with in the past, but now with a large population of motor units with more comprehensive and complex computational frameworks to understand how interneurons are affected based on the discharge profiles of motor neurons, because you understand the circuits of each motor neuron, I mean, somewhat. We are also interested in the initial discharge rate in fast contractions because that drives specifically the high threshold motor units. So if you want to investigate high threshold motor units, one way we can do that is to ask people to do contractions with the intent to do as fast as possible. So that it's another avenue of things we can try to explore and try to investigate specifically biomarkers on that. We also hopeful that we will be able to kind of move forward with this with the effect of resistance exercise and lifting weights on the motor neurons of people with motor neuron disease. We showed that in older people, we were able to rescue a lot of the excitability that they have with resistance training. So there's some data on that could happen in ALS too, in some animal models. So that's something that we hope we'll be able to do in the near future and use all these techniques into a more applied exercise way. And well, maybe in the future that's a bit away. What we'd like to do is then to have a lot of data, right? So if you have large data sets of motor unit firing profiles for people with MND, we can really use these firing patterns to inform in silico modeling. So you can use computer simulations. If you understand how they work, then we can restrict the solution space to say, only model in a way that we know the outcomes we want. And that could help us to understand the synaptic organization of how your brain is trying to drive those motor units, which would help to untangle a lot of things on how we understand the motor neurons are working. If we manage to get a model that works and then that is good enough, we can use that, new model to then simulate possible treatments. So you can change, parameters of the model and simulate treatments. So a lot of treatments could be trialed even before they go to people. We could trial them in silico first and have an understanding why they, could work or not, or what would be the consequences I would expect from them. We could model or simulate specific mechanisms of the disease. You could simulate different disease progressions or change parameters and look at different progressions of disease phenotypes. And maybe even if we get enough data for a specific person, we would be able to create patient-specific models that could help to figure out a lot of the heterogeneity of the disease. So, having large data sets of firing patterns of motor units could really help us drive all that via all the technology that we've been seeing emerging now these days with large data sets and computer modeling. That takes me to the end of my talk. So I would like to acknowledge, the participants and the families for the time and for helping us with our research. All the people at the Royal, all my team that has helped me with the research, FightMND for supporting us, MND and Me Foundation, MNDRA Australia. So thanks for everyone that has helped us. I'm happy to take any questions if anyone has any.
Great, thanks so much, Gabriel. That was really interesting to learn about the different types of neurons, and I found it really particularly interesting to know on a molecular level what the benefits of resistance training were. So yeah, that's really cool to ask. Yeah, it's we've got time for some discussion and questions. So if you have anything that you'd like to ask, please use the reaction button to raise your hand at the top there, and I'll be able to allow your mic and camera. So Peter Crouch, we've got a question from Peter Crouch. Please go ahead, Peter.
Yeah, g'day. Thank you, Sarah. I cannot see how to turn my camera back on, so I'll go
Right.
sans camera. Sorry about that. Um, uh, Gabriel, thank you very much for the presentation. I really enjoyed that very much. Fantastic work and really important work in particular. You mentioned that your delta F did not correlate with the functional rating scale. I cannot recall if you said that the maximal discharge readings you were getting correlated with the functional rating scale.
It did.
But it did?
Yeah.
Okay. So I guess my question is, with the delta F readings, what was the limitation with the correlation? Because you showed that the delta F did change over time. The FRS does change over time. So what was the limitation with seeing a correlation there?
I think possibly that the changes with the maximum firing rates were larger, right? And possibly less variable in comparison with the delta F. So with the delta F, we had, there was a bit more variability longitudinally. So you had some people that went up and then went down, right? While with the maximum discharge rate, it was really a trend more clear that, most people, the majority of people went down as time goes by, which then clearly mimics what the clinical measures show us, right? While the
Yep.
delta F, it's difficult, like, because if we assume that there is a possibility that there is a transitional increase in excitability before it goes down, we might think we're looking at everyone at the same time, but in fact, we look at people at different times of their progression. So while some people are going up before going down with this specific measure, they might mimic it, other people might be going direct down. So I think also a lot of the variability of that might be capturing it. And I don't think the functional scale won't reflect hyperexcitability, right? Because it can't. It has a ceiling, so it can't go up. While if these measures are going up, that's probably a lot of the signal will be lost to noise, I guess.
Okay, so now I'm going to ask the question that I really wanted to ask, and that is, where do you think do you think there's capacity with the high density EMG to measure functional treatment response?
Yeah, absolutely. I mean, I think particularly for our preliminary results, the maximum discharge rate, it's a bit more promising now because we could clearly see larger changes compared with the delta F. I think we could improve those measurements, and if we can make them a bit less variable, and if we can control better things in between visits, if we can improve how we do these things, then we could probably decrease a little bit the amount of variability and noise and get better signal, and potentially we could look better at functional responses and treatment. I think it is possible, but at the stage we are at now, I see the maximum discharge rates as something that seems to be more clearly mirroring the disease progression.
Yeah. From a naive perspective with your EMG recordings, if you were able to measure a functional treatment response using high density EMG, would most of that signal be reflecting of a neuronal improvement or a muscular improvement?
A new road. I mean, so that's why I try to. So the first part of the talk, I was trying to refer to what we are measuring at the muscle, right? But
Yep.
these are electrical signals that come from a motor neuron,
Yep.
right? So, and that has a one-to-one relationship. So although we're measuring at the muscle, the electrical signal has the only source that it has is a motor neuron. So every change we measure in terms of electrical activity, there are some parameters of the EMG and high density EMG that tell us about the peripheral muscle part of component. But typically the discharge rates, they are the source. They are, I'll probably say 100%. They are most largely the neural input from the motor neuron. So these type of measurements we do, they are largely central neuron, central nervous system sort of signal. There are other things we can measure with high density EMG that would tell us maybe a bit more about the conditions of the muscle. But these ones are largely driven by motor neurons. So that's that's one of the main things I'm trying to kind of bring it up, is that although people might think of EMG as a muscle thing, really it's a window into our nervous system.
Right. So you measure neuroprotection, but not muscle improvement per se with EMG.
Well, we could measure muscle improvement with if we can measure force together with EMG, which is something we do, normally here, then we have, the mechanical component of it and then the neural component of it. So like we can have indication what muscles are changing or not based on force measurements too.
All right, cool. Thanks for that. I'll stop holding the microphone.
You're welcome. Thanks for the questions.
No worries.
No dramas, Peter, at all. I just have a question from the chat from Davor. He says, on longitudinal studies, the numbers are lower as the studies progress over time. For example, 5 at the longest time point. Can you give any insights on the reasons for this?
Well, with disease progression, it gets really hard to keep recruiting people. So we've tested people at the Royal when they come for the clinical visit. So a lot of patients, they stop coming from the clinical visit. They are typically around the 3 months mark, but of course, some of them might get delayed. So 5 visits might mean probably a lot more than 1 year. So yeah, it's the typical challenge in this type of population to try to keep recruiting them for the test at the hospital. As the disease progresses, many of them stop coming. They start to go into telehealth or online consultation with a neurologist.
Yeah.
Some of them lose function and we can't get signal. The motor unit quality deteriorates, and there are all these difficult parts of it.
Yeah, it's definitely a challenge. I have another question from Gethin in the chat. Is there any data on whether people with different proportions of fast versus slow twitch muscles are differently susceptible to developing MND, or if it affects the rate of progression?
That's a great question. I don't have the answer for it. I don't know. I don't know if anyone has done. I mean, it's a complicated study because you have to do biopsies and like get muscle samples and typology of muscle fibers or myosin heavy chains, and those are complicated studies. You have to sample a lot of people and then wait to see if or you could get samples from patients, I guess, too. I don't know if people have done that specifically. My understanding my suggestion that the higher threshold units deteriorate are mostly come from animal studies, I guess, where people try to control that better, and that typically they find faster involvement of higher threshold motor neurons or fast twitch fibers.
Yeah. And he also asked whether EMG could provide data to help inform the die forward versus die back hypothesis of motor neuron death.
That's a good question. Well, if we could get to a point where we have enough data, then we can, as I said, reverse engineering, go back and understand what are the central commands we are sending to the muscle. Then it is possible that we can try to tear apart, how much of that signal we can understand that's coming from upper motor neurons, and then how much of that represents, lower motor neuron pathology. That's one possible way to go about it, and it is possible. We are working on it. Another way to work on it would be to integrate TMS studies. So we could do TMS together with high density EMG. We could then, figure out and do all the measurements that are meant to be cortical, and then try to tear apart whether, if we do the measurements at the same time as they progress, whether we can have enough data that will tell us information about upper motor neurons and lower motor neurons and working together. So that could be another way we could think about it too.
I just have a curious question about what participating in this study was like from a participant perspective. You talk about it being a low non-invasive measurement. So, what does that look like? What kind of electrodes go on the person? Like, where do these electrodes go? How long are they needed? Do they need to do anything while they're being measured?
Yeah, And yeah, that's my fault because I was so stuck in like not going overtime. So I kind of cut a little bit of that type of information. So typically they would look like, you know, they would come from the consultation. Then after they start participating in the studies, they would sit on a chair, and then we would put an electrode like this one, as you can see here on the screen. I don't know if you still can see my screen, on the top of their tibialis anterior, so the muscle in front of your lower leg, so in front of your calf muscles, in the front part of the leg. Then you connect to the computer, and then we would ask them to do first a contraction and measure their maximum, you know, electrical activity of the muscle. We then go and ask them to do this, what we call triangular contraction. So they will go and make the electrical activity of the muscle go up, and then they make the electrical activity of the muscle go down. They can see that on a screen, and we give them, you know, kind of a target, and they try to follow up with that target. So it would take, you know, probably around 10 to 15 minutes for them to do. It's non-invasive because it's just a sticker electrode that kind of is placed in the top of the muscle belly, and they have to do a few maximal contractions, and then they have to do a few submaximal contractions, which are around 40% of their maximum capacity.
By a contraction, you mean to sort of move your leg?
Yes, sorry. By a contraction, I mean an isometric contraction is like they try it against a resistance. So we hold their feet, so we keep their foot non-moving, but they try to overcome the resistance that they won't be able to, will be static, right? But they will produce force against a rigid device.
Yeah. Okay. Thanks. I just have, I'll just do one final question from the chat from Sally. And she asks, do you have any thoughts on why we might see preferential wasting of intrinsic hand muscles when they should have similar recruitment thresholds?
So, yeah, and that's an interesting one, right? I mean, it could not necessarily be all. So hand muscles, I don't remember from the top of my head now, but I don't know why I think like the recruitment threshold of hand muscles might be lower. So they have larger representation on their cerebral cortex, right? So they might have more input from upper motor neurons. So if you assume that, you know, like everything started at the upper motor neurons, and they receive more projection from upper motor neurons, and that could be one of the reasons. We haven't done a lot of study on hand muscles. They are small, and the electrode grids, we have smaller grids that could be placed on hand muscles, but they could be a bit more challenging with the decomposition. From the recruitment threshold perspective, I don't remember off the top of my head if the recruitment is different or not. But yeah, I think it's more related to the amount of, you know, cortical input they received compared direct monosynaptic projection input they receive from the cortex compared to the other muscles, I guess.
Okay, thanks. Well, I think given the time, we might wrap it up there. If anyone else has anything they'd like to ask Gabriel or follow up in terms of anything he's presented today, I'm sure he'd be happy for you to reach out to him. Or if you'd like, feel free to reach out to us at FightMND, and we can help connect you. So thank you so much for taking the time to speak to us today, Gabriel. It was a really interesting talk to hear about this different perspective of biomarker design in a sort of non-molecular, non-invasive approach. So thank you everyone for joining us today for the April FightMND Scientific Seminar. We hope you enjoyed the presentation. Please do join us again next month when we will have an exciting new seminar. Keep a lookout in your emails for news on this seminar, which will take place on Tuesday, May 26th at 12 p.m. Australian Eastern Standard Time. So thank you again, and we'll see you next time.
Thank you. Bye.
Thanks, Gabriel. That was really fascinating, and I think you explained it so well.
Thanks, Sarah. I tried to spend a significant amount of time trying to make sure people will get through the parts that are not familiar to everyone else.
Yeah. I think our care grants manager is going to reach out to you because she thinks some follow-up studies might really